Good afternoon, everyone. I have been asked to stand here, used to standing behind the podium, so that the folks on the live stream can see better. So welcome. It is truly a great pleasure to be here and to welcome you all for this Don P. Giddens Inaugural Professorial Lecture. And in this IPL, we are honoring Michael Schatz. Michael joined us as a Bloomberg Distinguished Professor in the Department of Computer Science in 2016. He also holds appointments in the Department of Biology and in the School of Medicine's Kimmel Cancer Center. He is a globally recognized leader in large-scale computational examination of DNA sequencing data, and the algorithms, computational systems, and machine learning techniques he has developed have very broad applications in areas that include agricultural genomics, comparative genomics, human genetics, and personalized medicine. And amongst his accomplishments are the algorithms he developed that advance our understanding variations in thousands of human genomes. This knowledge can uncover the genetic basis of cancer, autism, spectrum disorders, and other major diseases. Michael is also very committed to seeing that his discoveries have direct impact on human lives. Just last year, he and a team of researchers developed open source software that promises to revolutionize how DNA is sequenced, not only making the process faster and less expensive than ever before, but also making it possible to conduct genetic tests and deliver diagnoses on a portable device outside of traditional healthcare settings. And so I'm very, very glad that we are able to honor him, to celebrate his achievements, what he has done and what he will do. Now, before I hand the floor over, I want to also note that this event honors one of my predecessors, Don Giddens. He was the 5th Dean of Engineering at Johns Hopkins, where he also was a professor of mechanical engineering. His career has been defined by his commitment to research and education, and so it is truly an honor and a pleasure to be able to recognize his contributions to this school through this named initial professorial lecture, one of my most favorite events, actually. And with that, I'll hand the floor over to Randall Burns, the head of the Computer Science Department here in the Whiting School of Engineering.
Excellent. Thank you, Ed. It is a delight to have this event, and I'm here to introduce Stephen Salzberg, Mike Schatz's mentor for this event. But I want to reflect on Mike for a minute by anecdote. So I stay in my swim lane and let Steve introduce. I teach a freshman seminar, and in that freshman seminar, I use Mike's recent work on Uncall as a way, which Ed hinted at, as a way to show the inappropriate power of software. And this is a case in which Mike's lab and team and his insight has made a piece of hardware that costs hundreds of millions of dollars to develop 4 times faster. And it just shows that the power of data and insight can lead to transformative changes that are incredibly rapid. And so I hold Mike out as that kind of computer scientist. When I talk to people about what it means to be a computer scientist, that this is what the field can aspire to. That type of rapid change and widespread change. I first met Mike in 2009 at Supercomputing. And Supercomputing is not a bioinformatics conference, it's a me conference. And I was overwhelmed by the depth of the systems work that they were bringing to bioinformatics. And that is representative of the work that Mike and Steve Salzberg, who I'm introducing, were doing at the time, and that they continue to do. So Steve Salzberg is my PhD advisor, which is probably his most notable outcome. In addition, he's a Bloomberg Distinguished Professor of Biomedical Engineering and Computer Science at Johns Hopkins. He is the director of the Center for Computational Biology. And a couple notes about things that Stephen has done. One, he was the head of bioinformatics at the Institute for Genomic Research. This was at a time of amazing impact. This is when the Human Genome Project was uncovered, or was sort of, I don't want to say resolved, it's not a solved system, but it was at the height of the Human Genome Project. He was leading the bioinformatics team at the most important contributor at the time. He's been a professor at Maryland, subsequently a professor at Johns Hopkins before. I like Steve as a blogger. So he writes a column for Forbes that interprets biomedical research, news, media to the public in a scientific way that is accessible with the right amount of humor and sarcasm. And with that, Steve.
Thank you, Randall. Thanks so much. This is not about me, though. Today's about Mike. So I could give you a history of Mike's achievements, but I think he's going to tell all of us about some of his work in the past, as well as his plans for the future. So I thought I would just tell a short anecdote just for a minute. So I first got to know Mike when we hired him as a programmer back at Tiger, what Randall just referred to, the Institute for Genomic Research, we all called it Tiger, back in 2003. And he started working on genome assembly right away. The landscape of genome assembly, and he'll tell you about genome assembly, but it's you basically put genomes together from little tiny pieces of DNA. And the software that we had at the time, sorry, my mask keeps slipping, but the software that we had at the time would take a couple of weeks to assemble a bacterial genome, which was a really small thing, but it was a big task. So you kind of get all the data all lined up, and then we'd hit the go button and let the assembler run for a couple of weeks. And then whatever it gave us, that was kind of it for us. And we would then use laboratory techniques to kind of finish the genome, which took many, many months. And so Mike, we had just gotten an assembler, I won't go into that story. We just got an assembler from another group that was much faster. And so I asked Mike to start helping us out with assembly. He's still working on that. And one of the first things I got back from him, I looked through my emails. I couldn't find this message, but I remember it. So hopefully it's not just something I created. Was he sent me these, so I said, try assembling whatever this genome was, some bacterial genome. And he sent me back like 15 different assemblies of it with all these different parameters. And it had not occurred to me at the time, because assembly was such a big test, but we had this faster assembler. And so he realized before the rest of us realized, like, no, this is just a tool. You can start fiddling around with it, and you can get much, much better results if you fiddle around with it enough. And then the laboratory work, instead of taking many months, will take a lot less time. And that's how we've done assemblies ever since. We're still doing that. From there, of course, we all, my group all left Tiger back in 2005, and Mike left as well and became a PhD student at UMaryland. And after that, worked on genome assembly for his thesis, as well as some other problems, including some problems on high performance computing. That's how I just learned he met Randall way back then. And then went on to the Cold Spring Harbor Laboratory, which is a world-class genomics and genetics research institute up in Long Island. And they loved him there. I'm on their advisory board, so I know that they loved him. Because after he left, they kept saying to us on the advisory board, can we get another faculty member like Mike? We want more people like that. And I said, it's really hard to find people like that. So he's one of the things that I've always enjoyed about Mike is the tremendous enthusiasm he brings to his own work, which he also communicates really well to his students. And you can see it in the way they present their work as well, which I've heard his students present many times. So without any more about that, let me introduce Mike Schatz to tell us about his current work and his plans for the future. Okay.
Thank you so much for the introduction. I think I have the lapel mic. I don't need this. Is that right? Okay. So thank you so much for being here. Thank you for the very kind words, the introductions. It's really a great honor. I'm like overwhelmed with emotion to be here. So like we just heard, so I've been in genomics now something like 20 years. So I thought we could take this opportunity to kind of reflect on some past work and then really talk about the future, about where the whole field is moving. So just to get it open, as you've heard, I've been in the field for some time, and it's really been a great pleasure to work with many different students, postdocs, collaborators, staff, just incredible environment. So I try to put up like this menage of people I've worked with over the years, and kind of goes on a little bit. And it's really, these are the people that I am here to thank, right? These are the people that have made me successful. I am here as sort of a platform to amplify their ideas, amplify their abilities, to focus them on the important and impactful science in the world. It's really their work that we're celebrating here today. So first of all, thank them. and thank all of you for being here today. I really mean this in a very, very deep way. None of this would have been possible without all of these great friends and collaborators. So as you heard, some of the problems I've been working on for several years now are pretty fundamental. So number one, like, what is your genome? We know inside of your body, inside your cells, there's this mysterious molecule DNA, and with it carries so much genetic information. Really fundamental problem is, what is it? Let's try to read this off. And then increasingly, what I'm becoming more and more focused on is like, what does it mean? What does it actually mean? What are all of those ACGTs in there actually do? What are they responsible for? How do we interpret them? Maybe how do we sort of modulate them in the presence of COVID-19 and other sort of environmental stresses? So it all starts with some sample. So in my group, we've worked on human genomes. Here's a sort of a cartoon from a cancer genome that we worked on. We've worked on agricultural genomes, major crop species to try to feed the world. We've worked on virus genomes. In fact, we participated in the COVID-19 response here at Johns Hopkins, trying to sequence viruses out of patients to really understand the mutations, get a hold of things like the Delta variant before it was recognized as being out there. So it all starts with these biological samples, and there's any number of reasons why you might be interested in them. And then what it takes is some sequencing. So these are instrumentation. Historically, I was hands-off with this, but as you've heard, we've become more and more hands-on, thinking about some of the special properties, and there's just sort of amazing things that can be done inside of that sort of technology. But then, out of these instruments comes very, very raw information. Little bits of DNA, little bits of ACGT, where that's where the algorithms, the computer systems, the machine learning, everything comes in to sort of assemble it, make sense of it, piece it together. And then there's a lot of comparative studies that could be done to find those associations with diseases, find other sort of traits, you name it. This is kind of the process as it works. So it's an interesting process. It's sort of, to me, very intellectually satisfying because it's very meaningful. It's a good computer science problem because there is a right answer, we think. So we can try to work harder and harder to engineer those solutions. And it just sort of, is just really meaningful in lots of different ways. Now, genomes have been assembled. The first complete free-living organism was actually at Tiger in 1995, a small microbial genome. But by far, the genome that gets the most attention is the human genome, right? So this is an ongoing project. It really started in the 50s, I would argue, when the structure of DNA was first determined. People were really interested in what that ACGT composition was. There's great interest to actually go out and sequence it. So that was in the 50s, and then as the technology matured, it became actually feasible to actually assemble, put together a human genome for the very first time. So there's a lot of efforts of big international work to make sense of this, led by a couple of folks, one being Francis Collins, who's now the current director of the NIH. Another gentleman, Craig Venter, had a private effort to sort of sequence the human genome. And this all kind of came to fruition in sort of around 2000. There was a press conference at the White House. President Bill Clinton at the time remarks, without a doubt, this is the most important, most wondrous map ever produced by humankind. And I just really like that sentiment. It was incredibly important. In 1,000 years, people remember this is the era that human genomes were first sequenced. It was most wondrous because it just opened up so many possibilities to understand our molecular makeup. And I really liked that it was produced by humankind. It was sort of built by many people, all contributing sort of ideas, sometimes even DNA. It's really, really, it was a resource for the world. So without a doubt, it is arguably the most important piece of biomedical data ever produced, be able to construct the human genome. But here's the twist. Here's the twist. It's been celebrated many times as being done and complete and being a tremendous resource, but it's not. This is the twist. The twist is certain parts of the genome today are incomplete, they're incorrect, they have mistakes, or just complete holes, right? So when we think about DNA, it's ACGT. If you open up the current human genome today, it actually does not start ACGT, it starts NN, meaning I don't know. It does that for millions and millions of nucleotides. It's a big pain, it causes all kinds of confusion, it means that we're gonna be missing a lot of things, there's just lots and lots of problems because of these big holes inside of the human genome. So why does it matter, right? We can't, so it's more than 100 megabases, which is about the size of a human chromosome. So it's like an entire chromosome has been missing. We can't study it very carefully. We're going to miss any variation. There's going to be all kinds of artifacts, all kinds of challenges. And then we just don't know what we don't know. Like, who knows what's in there? We know a little bit about some of its properties. We know it's really important for, say, replication, so that cells divide. We need sort of accurately to make copies of DNA, but we just don't know what we don't know. And it's really taken so long, right? Technology to sequence DNA has been in development for many decades, but it just couldn't be done because it's so complicated. And sort of more technically, it's just very highly repetitive, very complicated structure that the technology just really hasn't been possible to make sense of. Until very recently. So one of the main, I would say, thrusts of my lab over the last decade has been working with new sequencing technologies that can sequence DNA, make sense of DNA in very specialized ways. So today there are two platforms, one made by the company Pacific Biosciences, and another from the company Oxford Nanopore, that have these very, very special instruments that can look at DNA in ways that just previously weren't possible. The key trick for this is they're single molecule. They isolate single molecules of DNA. And then from that one molecule, they can actually read off the A's, C's, and G's, and T's. And no other technologies could really do this previously before. I will say these technologies were challenging to work with. We've had things like electrical problems where if you would touch the instrument, it would electrocute you. We've had issues where it caught on fire at times. And then at the sort of data quality has been extremely challenging. In the early days, the companies themselves admit the data were rubbish getting started, just incredibly erroneous, just not really great information to work with. But we knew that someday having this special sequencing would let us do special things and look at things in ways that have never been seen before. So we knew that it was a good investment in time, even though the technology when it first came to market was, in their words, rubbish, trying to work with it. And I'm very proud to announce today at my IPL that we've been successfully able to use these technologies to actually complete a human genome. I can't take full credit for this. In fact, I owe a tremendous amount of credit to Adam Philippi, who's a great friend of mine for 20 years back to Tiger, and also Karen Miga, who sort of spearheaded an initiative called the Telomere to Telomere Consortium to actually assemble a human genome for the very first time. To fill in all those missing bits, actually complete all those erroneous sequences, and actually make sense of the genome for the very first time. So it's an international consortium. There's many people involved, and I want to tell you a little bit about some of the things that we've been able to do to look at this human genome for the very first time. So first of all, through these new sequencing technologies, through improvements to algorithmics, and to improvements and just sort of manpower to look at it and study it, there is now a complete human genome sequence that you can download for the very first time. There's a whole series of papers. I'm going to be referring to this BioArchive thing, which is, if you aren't familiar with, it's sort of pre-publication, sort of disseminating the scientific research before it's completed through peer review. So I'm very pleased to say there is now a complete human genome. We added about not quite 200 megabases to it. It just fixes thousands of errors, it just wholesales improvements across the board. Special credit here to Sergey Nurk, who developed some of the key algorithms, although some of my lab members and many other people at Hopkins really contributed deeply to this assembly. So now we have this new sequence, and it adds all this new information to it. And one of the things that we really want to study is like, well, what's in this new sequence? And how do other people's genomes relate to this new sequence? Like, what's going on inside of there? And there's a variety of working groups that were set up to kind of look at it. And I worked in one sort of studying how this improves the analysis of human genetic variation. There's a preprint describing my work. Co-first author by my postdoc, Sergey Aganov, Stephanie Yan, I think who's here, a student of Rajiv McCoy, Daniela Soto, who's a student of Megan Dennis out at UC Davis, my student Melanie, I don't know if she's here, and my other student Samantha in computer science department. So we had this genome, and we really wanted to look at it and really ask the question, especially in these new regions and maybe across the whole genome, how would this really impact our analysis of human genetic variation? I think, when we went into it, we thought it would have some impacts on, at a very basic level that, there's new sequence, of course, that'll help us see things. But I don't think we really appreciated just how deeply impactful this work would be. So we wanted to look very broadly. So what we did was we looked at a large collection of human genomes called the 1,000 Genomes Project. They call it the 1,000 Genomes Project, but it's actually 3,202 samples. It's the way they named it. It comes from 26 populations from around the world. So kind of highlighted on the world map are these individual populations, each with a few hundred samples. So in collection, 3,200 samples. It turns out just a few months ago, there was some very new sequencing of this collection at the New York Genome Center. So it was like high quality data. They did, the New York Genome Center had done this analysis using the old genome. So we could kind of directly ask the question, well, how would it look looking at our new genome sequence? Now the challenge, and this goes back to supercomputing, was that it's 3,202 samples, each 30 odd gigabytes or so. So it's about 100 terabytes of input data. The peak data footprint will be many petabytes, millions of core hours to actually look at this. So it's a nice supercomputing type challenge to actually look at this. When we started the processing, we actually did it here at Marcy, the Maryland Advanced Research Computing Center, try to use the Hopkins resources. And it was working, but we realized it was going to take a really long time to go through. We were really worried about running out of storage and sort of things that computer science really worry about. Well, it so happens, in addition to sort of working at Marcy, I am very deeply, in fact, I'm the co-leader of a major project from the NIH, at the NHGRI, to start something called the Genomic Data Science Analysis, Visualization, and Informatics Lab Space. I did not name it. We call it the Anvil for short. And the idea is that in biomedical research now, there's huge amounts of data being generated. It's very complicated. It's very expensive to have sort of data centers at every university, at every hospital set up independently to kind of look at these data. So wouldn't it be amazing, instead of having to copy data all over the world many times, what if there was sort of a centralized resource that everyone remotely could connect to, sort of take advantage of the same tools, do it at huge scales, and also do it in a secure facility so that if we're looking at patient data, we can do so and make sure that it's protected. So that's the Anvil. It's a federated system. It's composed of many sort of components that can do interactive analysis or batch computing or visualizations. There's lots of cool work behind the scenes. Critically, though, it runs inside of the Google Cloud. And critically, we have technologies in place where we can orchestrate very large workflows so that if you have thousands and thousands of samples, we can go through it very quickly. So my student Samantha wrote a workflow. This is just sort of a little piece of it. It starts with this raw sequencing data, just raw ECGTs, and then does lots of comparisons to human genome, this new human genome, so we can kind of make sense of all the variation that are present there. So this was a big compute. We were routinely running at like tens of thousands of cores for several weeks, just trying to get through these data very quickly. So what did we find, right? So we knew that we were adding about a chromosome's worth of extra sequence data. And it turns out we add about a chromosome's worth of extra variant information. We add about 1 million extra variants across this large population that had just never been seen before because they're in parts of the genome that were just really tricky to make sense of previously. So that was sort of the expected result, and that's amazing. It's taking data that we already had and making it more valuable, finding millions of variants that have never been seen before. There was another twist, which I didn't anticipate, but is also, I think, equally or perhaps even more important, which is very counterintuitive, and it's like warps my brain. I'm incredibly grateful to Rajiv McCoy in particular, who's like very patiently walked me through these calculations. So on a population scale, we see lots more variation, but on a per person scale, we actually see a lot less variation. And this is a very complicated, surprising result there. And this is just some of the results looking at admixed American individuals, several hundred here. We're comparing, GRCH38 on the left, CHM13. The big thing to notice is you get this big drop in the number of variants. And that's incredibly significant biologically, as to understand why that is. So there's a few reasons, and kind of to dissect it, we made a bunch of plots like this, where so for each of these samples, in this case, say there's 1,000 samples, we can ask the question, what fraction of people have a variant that's present there? We can just sort of tally up how often those sorts of variants occur. This is so-called allele frequency spectrum. And when you look at it, by and large, when you compare GRCH38 to the CHM13, they're pretty similar, but there are certain regions where they're quite different. And those are the most interesting regions to dissect. So on the right there, there's a region where they're quite different. We dig into it and we find things like this. So on the left here is sort of a cartoon version of what's happening on GRCH38, where the reference genome itself has some particular nucleotide, let's say A, and then all or most of the other samples have some other nucleotide there, let's say T. But when we look at sort of the corresponding region in CHM13, it sort of flips the mirror, if you will. The reference genome now is a T. All the samples there are also a T. So there's no variant at all. So we get lots and lots of occurrences where the reference genome itself has this like very rare or very likely to be erroneous sort of nucleotide present. So when you compare to other samples, you just sort of get these artificial false positive variants. So that's bad news. Bad news number one. Bad news number two comes in the middle here, where there's a bunch of variants that have allele frequency right around 0.5, which is like just a weird place to be. And the sort of interpretation, we have lots of other data kind of confirms this scenario. What we think happened is the truth in real people's genomes and real nucleotide sequences. There are two segments that are very similar. And it turns out that CHM13 gets it right. There are two sequences that are very similar, but they have, like, say, one difference, where in the similar regions, maybe there's an A in one, there's a T in the other. But in GRCH38, they're collapsed on top of each other. This is like the most common challenge in genome assembly. So that when you're matching up sequencing data from other individuals, you get sort of a mashup of the two sequences, and then you get about half of those positions will be A and half those positions will be T. And this is really, really bad news biologically, because suddenly we're going to be systematically making all these errors when we're trying to interpret all the variants that are present there. Very bad news. We also see similar things using long read data. This is led by my student, my postdoc, and this time I'll go through it. But again, we're seeing really sort of bad things in GRCH38, the existing human reference genome. They're systematically improved in CHM13. So we're excited about this new genome. It's cleaning up a lot of the variants that are there. One of the major analyses we're due was to sort of dive into those regions that we put together for the very first time, and then just ask, what are the makeup of those variants? Because we're looking at so many populations, we could actually sort of systematically check and see, well, how common are they in different parts of the world? Thanks to Rajiv and Stephanie and their group. And we do find certain variants that are highly, highly enriched in, say, just Central America or just in Africa. And that's what we're kind of investigate now, like how do those variants relate to different phenotypes in different parts of the world. It's just really an incredible opportunity for biological discovery that is made entirely possible to this new reference genome. And then finally, as we're sort of digging into it, we asked the question, well, there are certain genes or certain variants that are known to have clinical impact. They're known to be associated with like different diseases. And we could ask, very sort of critically, compared to GRCH38 or this new assembly, how does it improve variant calling? And what we were able to measure is sort of whole scale improvements on both false negatives and false negatives. So we can just get much more accurate clinical variant calling inside of these genes using this new reference genome. So just that was sort of a whirlwind tour, but what we're trying to demonstrate here is through this new reference genome, we're getting huge improvements to our ability to analyze variation. This is at a global population scale, also for clinically relevant genes. There's been many builds of the human genome. The current one is 38. Sort of internally, we call this like GRC 39. It hasn't been blessed with that designation, but in my mind, that's effectively what we've done here. Now, there's tons of people that were involved in this work. I'm super grateful to all of them. There's a whole series of different preprints that are currently under review right now, kind of describing all these results in more detail. I'm happy to talk about this, but I wanted to also spend 5 minutes talking about what's next. So just bear with me here. So in some regards, what we're doing has been part of our culture for a very long time, right? The technology is getting smaller and cheaper and faster, and we're able to carry it in our pocket. Like, oh, well, I happen to have a DNA sequencer in my pocket. So it is, we're really getting to this point where these tricorders are really possible. And I think it's really exciting to think about, now that we're all starting to carry tricorders in our pocket, how are we going to be able to use them very effectively moving forward? So there's a number of sort of scientific and engineering challenges that I think need to be addressed here. The first is really, really fast, really, really accurate, just really ubiquitous sequencing. I think there's been a lot of improvements there, right? Most clearly demonstrated through this T2T assembly, right? That has no unresolved bases for the very first time, just universal improvements. It's amazing. Now, today you cannot actually take this and plug it into my iPhone, but they're working on it. On the right there is sort of a screenshot on Twitter where the CTO of the company is sort of demonstrating that it has a funny name, the Smidge ion, that they hope to someday to actually really directly plug into the iPhone. I think it's going to be transformative, right? Because suddenly we can have a diagnostic in our pocket. I can think of no better example than COVID-19. You get the sniffles. Is this something I should be worried about, or is this just a common cold? Well, what if you could just sequence the genome at home, tell you, right away, this is something to be worried about. In addition to sequencing genomes, I'm really excited about other sorts of sort of biomedical data. The sort of next on the list would be so-called epigenetic data, looking at chemical modifications that can occur on DNA. I'm also very interested in integration with like healthcare data, imaging data, environmental data. I know it sounds crazy, but wearables like smartwatches, I think are going to be incredibly important in the future. It's just this continuous physician's test, constantly monitoring you, constantly looking at your activity. It'd be hard for you to notice, I don't know, a 10% change in your respiratory rate, but something like a smartwatch could detect that right away, and that could be a really important biomarker. So I'm really excited about that. We've started to apply this in disease settings. So using some of these new technologies, we're able to see classes of variants that have never been seen before. So here is a variant in the BRCA1 gene, the BRCA1 gene, kind of the most substantial risk factor gene for breast cancer in a breast cancer patient that had gone through diagnosis using sort of traditional approaches, didn't see that there are any variants of concern, but now using these new technologies, we can start to see them. So I think there's a whole aspects of the genome that are completely unexplored, and now finally we have the power to go look at them. So a lot of this talk has been focused on human genetics, but as you heard in the beginning, I also do quite a lot of work in agricultural species. About a year ago, we had a major study looking at tomato genomes, where we're using these same sorts of technologies. We could find variants associated with like the fruit size, the taste, the flavor, the growth, tremendous results there. Just on Friday, we had a new paper looking at this model system, this actually beautiful flowering plant called Arabidopsis, where we could look inside of the centromeres for the very first time to be able to see some of the amazing dynamics that are there. So I think we're going to see these sorts of technologies, yes, in human health, but very, very broadly across the tree of life. So the next challenge, and this kind of speaks to Randall, is this work is, yes, Hopkins is a pioneer, and yes, there are certain institutions that are really obviously significant to it, but it really, really needs to be a worldwide endeavor, right? Again, COVID-19 being this great example where viruses and pathogens halfway around the world are really, really relevant in our backyard. We don't want to be doing this in isolation, in a silo. We need that integration of data at extreme scales. I've been arguing, and I've been working with Ben and Stephen and others for years, that cloud computing offers a lot of advantages for this. And it's kind of amazing to see this actually come into fruition. Now there is finally this platform that we can start to take advantage of. But there's, as much as I wish, the cloud was a silver bullet, it brings new sets of challenges. Probably number one is that there will not be a single, I wish there was, but realistically, there's not going to be just one cloud where all of these data exists. There's going to be many sort of clouds set up to do different things. There may be legal or privacy reasons why they can't talk to each other as deeply. But nevertheless, we can build on new interoperability technologies to make sure all these different systems can talk to each other and work together as well as best as possible. So there's sort of the way I think about this, and this is an analogy I came up with Adam Philippi a few years ago, is the way we understand the weather in your backyard is by having weather stations distributed all over the world so we can see the fronts, so we can see these patterns emerge. Well, this is exactly what we need in the case of genomics. We need these sorts of instruments all distributed throughout the world now that we actually have this technology, so we can anticipate some of the pathogens, other sorts of variants, other sorts of mutations, and other exposures that are all around us. And then finally, these are great, amazing technologies. We have great computing, but we actually have to pay attention make sure that this is equally available for all. And I would say genomics historically has done a very bad job at this. On the left here is a sort of a breakdown of samples that have been studied looking at different disease associations through something called the GWAS catalog. Probably can't see it in the back, but that huge blue space across the top is about 3/4 of the samples that have ever been studied come from European donors. And then this other 1/4 is sort of just little slivers of other populations. And this is a big problem. This is a huge, enormous problem. And especially because today, a lot of the sort of information we get about genomes comes through statistical approaches, where you're applying machine learning or statistics to sort of model what you observe, look for patterns. But if all of your data come from one ethnicity, this is going to be very bad news because those results may not generalize. We could be making life or death decisions, and it could be horribly biased. So there's just a bunch of there's just many interventions we need to do to improve this inequality to make sure we really pay attention to it. Top of the list is, we need to balance out the pie chart. We need to collect data from many different populations, make sure it really is a comprehensive look at genetic diversity across the whole world. There are things we can do at the software level to detect such biases and maybe try to correct for them, statistically, although obviously better data would be all the more important. Pay attention to it at the level of experimental design. So it's not just an afterthought, but something we put really up front and center. And then finally, especially here at a great institution of education and learning, let's make sure that the next generation is really poised and ready for this. So I'm proud to say in my own class, so here's my student Samantha teaching my undergraduate class about genomic analysis in the cloud, where they're actively building pipelines looking at that new genome. is part of their classroom work is to do this sort of research that's never been done before. And I'm very happy to do this at Hopkins. This is why I'm here. This is one of the great things about being at Hopkins, where we get to work with such outstanding students. But if we can look just a little bit beyond that, I'm also very deeply involved in something called the Genomic Data Science Community Network, where we're making a very sort of strategic effort to go beyond the usual leaders in genomics, the usual sort of suspects, and reach out to those that have historically been underserved. So we've been reaching out to historically Black colleges, minority serving institutions, tribal colleges, community colleges, to build up this network of faculty that are there to really trying to make a difference, really trying to lift up everyone, lift up all their students, lift up everyone to be able to take advantage of these genomic data. So to get started, this has been great work with Jeff Leek, who's here, and Fred Tan, and Carrie Wright, Ava Hoffman, and several others around Hopkins. We've reached out to about 25 other faculty of these other institutions. This is something I'm super proud of. We're trying to work with them to develop training materials, provide access to data, provide access to compute, so they get equal opportunity to all these resources as well. So we're kind of writing up a position paper right now. We're trying to get them engaged in sort of all the regular meetings. We're trying to develop teaching materials and curriculum that they can share amongst themselves using things like Jupyter Notebooks and also Galaxy, which I'm now involved in. The goal is, let's sort of build this network of educators that can all work together. But I would say Hopkins, we really need your help. And I know Jeff and I, we're really excited about this, really passionate about it, but we need help to make this go to the next level. So we're really on this cusp of this biotechnology revolution. I really do see that these tricorders are coming for all of us. I know that sounds crazy, but 20 years ago, it was probably crazy to think that we'd all carry professional digital cameras in our pocket, but we do today. We're kind of right on this cusp. There's endless applications to learn more about ourselves, improve our health, study the world around us. But there's going to be a lot of hard work that has to go into it. So over the next 20 years, so Genomics 2041, I think we're going to be sort of moving from really, really deeply into this sort of analysis of what do genomes mean, moving more and more into the interpretation. And the way we're going to make progress there is through this whole stack of technologies. We have to think about the sensors, we have to think about the computing systems, we have to think about the algorithms, we have to think about the machine learning. And at the top of this great pyramid, that's where we're going to make these discoveries. That's where we're going to be able to integrate across all these different lines of information. Now, this work is incredibly important, but it's really currently out of reach for many people. So we need to make and take immediate action to ensure that we have equality for all. And then if I'm honest with myself, this is going to take a lot more than 20 years. In 1,000 years, there's still going to be people pondering the meaning of the genome. It is so incredibly complicated, and it's so incredibly context specific. When you have to think about the environment you're in, the exposures you have, the interactions you have, all of that really, really plays into interpreting genome. It's incredibly difficult to predict. So there's a huge number of people at Hopkins and beyond that I want to link for this. So here I have kind of a listing of my sort of lab members, great collaborations across Hopkins, the Bottle Lab, Langley, Leek, McCoy, Salzburg, Taylor, and TIMP Labs, other sorts of great collaborators all around the world. Funding through this has been from the NHGRI, the NCI, Mark Foundation for Cancer Research, NSF. And what I'm most proud of is also through the Bloomberg Professors Program. Before I close up, I would just also like to take a moment to have a moment of memory for James Taylor. He's a great friend and colleague of mine that passed away about a year and a half ago, but I think about him every single day. A lot of the work he did really set me up, set all of us up to do enormously impactful things. And I want to take advantage of this opportunity and just wanted to thank him here at this IPO. So that, I really thank you again. I'm happy to take any questions. It's really a great honor to be here, and thank you all for coming. Thanks, everyone.
Thank you very much, Michael. And questions from the audience? Randall.
So I'd be happy for you to disabuse me of this opinion. But when I look at the advancement, I see lots of improvements in data quality, lots of improvement of computational outcomes, but not the same order of magnitude change in the amount of computation per base pair. You're investing an incredible amount of computation, it's more accessible, but it's not reducing by factors of 10. Is there a pathway where we can reduce the amount of computation needed to interpret data?
Great question. I think, well, orders of magnitude are always hard. I think those are Nobel Prize discoveries in themselves when you can make such an advance. I think there have been some advances there. Things like better indexing, sketching and sampling techniques. I know Ben teaches a whole course on this. I'm very interested in this intersection between computer systems and machine learning. There are these technique called learned index structures, where you take sort of classical data structures, classical sort of approaches, and you turbocharge them by taking advantage of in real world data. There are these intrinsic patterns. If we can model those patterns, we can sort of turbocharge these searches. I'm really excited about these as sort of a general class of algorithms, a general class of ideas that can turbocharge genomics very broadly. So I agree. One option is just throw more hardware at it. Another option is think harder, think deeper, and we're trying to do both. And I think both will be necessary.
Yes.
I was interested in what's been learned about autism through genomics.
Sure. And so this goes back a few years, especially when I was at Cold Spring Harbor Lab. I helped participate in this very large study where we sequenced, it was more than 2,000 kids with autism spectrum disorders, their non-autistic siblings, and then also their parents. And then we were just sort of broadly asking the question, what was special, if anything, in the genomes of kids with autism compared to their family members? And it was a really meaningful project. And the main result there, and actually since this work, there's been other sort of follow-up studies that have confirmed it, is recognizing the importance of what are called de novo mutations. So your genome and all of our genomes are largely made up of our parents' genomes, and their genomes are made up for grandparents, and sort of gets passed from generation to generation to generation. But in that process, right, your genome is 3 billion nucleotides long or so. Actually, double that because you have maternal and paternal. Occasionally, once in a great while, there'll be this sort of spontaneous change or mistake in this replication process, where there'll be this so-called de novo mutation appears. We all have them. Everyone has about 100 genome wide. And most of the time, right, the genome is quite large. So if you only have 100 of them, most of the time they have effectively no measurable effect on anyone's sort of behavior. But occasionally, those mutations may occur in gene sequences. And occasionally those mutations may occur in genes that are really, really central to like neuron development or basic biological processes. So the main result of that research was when you look at the genomes of kids with autism compared to their non-autistic siblings, they all had de novo mutations, but in autistic individuals, there was a much greater rate of these de novo mutations that were disruptive to the genes, right? We all have them in more or less equal numbers, but in kids with autism, they tend to be quite disruptive. It's still mysterious, is there something that is mediating that? Is it totally random? We try to associate it with other sorts of behaviors, other things. But today, the main result is just the occurrence of these de novo mutations. Yeah, there's been a lot of research since then to confirm that.
One more question here. Yeah, Jim.
So my question really has to do with data privacy. You've benefited enormously from access to all of this marvelous genomic data, and there's kind of no cost in a way, or no risk, right, to share it at this point. The game. Well, maybe a little bit of risk. And so my question is kind of how do you think about this, right? Sort of in your picture, 20 years out, where maybe you found sort of connections between certain sequences and antisocial behavior or something. What do you see as the future of data privacy and genomics and society's best interest?
It's a great question, right? So there's this really fundamental tension, which is that a lot of the power of genomic data comes through data aggregation. If I just have one person's genome, it's hard to say too much. As we just saw, this is, one paper is looking at thousands of genomes. Today there are studies looking at, upwards of millions of genomes all at once. So we need all of those genomes to come together so we can sort of detect patterns. But you're right, as we're doing this aggregation, there can be a tension in the privacy considerations that are present there. I'll say for the cloud platform that I'm leading for NHGRI, we take security very, very seriously. All the data sets are consented. All the data sets go through an authorization process where they're just not, some of them are open access, but the vast majority of them, you have to apply for access, and there's a series of sort of safeguards in place there. Something I'm really excited about is there are kind of statistical approaches and even encryption approaches where you can do analysis on data, where you can make really strong guarantees about not revealing too much. So I'm excited about this in the future as a way where you can potentially share data without sort of being fully exposed. But at the same time, millions and millions of people have already done 23andMe, have already gone through MyHeritage. So I think there's just tremendous interest, very broadly to take advantage of these data. I think a lot of it's going to come through education and policies and legislation. But I'm interested in both technical solutions and some of these policy solutions.
Well, let's thank Michael one more time.
Thanks everyone so much.
And I would like to, over here where we can have a photo.
Okay.
So we have a souvenir of this event for Michael. So I'll hand that over.
Okay.
Congratulations. Okay. Congratulations.
Thank you, Ed. Thank you so much.
Thank you all for joining us. And those of you who are here live, as opposed to on the live stream, you can actually pick up an actual cupcake on the way out. And if you want to mill about and have it, enjoy it outside, please. The rules. But thank you all again for joining us. Thank you.
Thank you, everybody.